enzymes that are made. See, and you see this several times over the course of these reactions. So as this molecule becomes modified, release a little water, here's a reduced coenzyme right here. So you're going to remove electrons from this 6-carbon molecule. They are going to attach to NAD to become NADH plus H. So here is your reduced coenzyme. This is one of the most important products that you get from the citric acid cycle. Remove another carbon. Now you only have 5 carbons. You're going to reduce another coenzyme. You see the same thing occur. Take off another carbon, and over here you see you're going to generate one measly molecule of ATP, and this is just through substrate level phosphorylation. Continue to modify these carbon-based molecules. FAD becomes FADH2. Here's another redox reaction. Oxidize this molecule, reduce this coenzyme. Water comes into here, another reduced coenzyme right here, and you're back where you started, oxaloacetic acid. And now you're ready to receive 2 more carbons to become citric acid, and you can go through this series of reactions again. So you keep, we refer to it as turns of the citric acid cycle. With every turn of this cycle, for every individual acetyl-CoA that donates its carbons, or every individual pyruvic acid, you make 1, 2, 3 NADH plus H, 1 FADH2, and 1 molecule of ATP. This is just for one turn of the citric acid cycle, or the Krebs cycle. Remember that for each molecule of glucose that goes into glycolysis, you make 2 pyruvic acid. Going back to this picture on the left side, 2 pyruvic acid will make 2 acetyl-CoA. So here's 1 acetyl-CoA that's carbon going through, and then the second one will head into the Krebs cycle. So if you're asked for each molecule of glucose, how many of these products are produced, then you'd say 2, 4, 6 NADH plus H, 2 FADH2, and 2 molecules of ATP. So that's how much is produced per molecule of glucose. Remember, it is split to make 2 pyruvic acid. So you have 2 turns of the citric acid cycle per molecule of glucose. I hope this
for making alcoholic drinks is called fermentation. The reactant in fermentation is sugar. It comes from natural plant extracts, barley in beer making, and grapes in wine making. These sugars have the general molecular formula C6H12O6. Fermentation converts them into ethanol and carbon dioxide. You might notice this equation isn't balanced yet, so have a go as a mini challenge. Pause and continue when ready. Here's the answer. The problem is, this reaction happens far too slowly to be of use to industry, but it can be catalysed by the enzymes of a naturally occurring fungus called yeast. Let's move on to our second route. This one also makes ethanol, this time from a hydrocarbon called ethene. You might have come across it in our videos on hydrocarbon cracking. Here's the ethene molecule and its molecular formula. Here's the ethanol molecule from before. Now look at the numbers of each atom on both sides. Can you think of a very common molecule we can add to ethene to produce ethanol? Again, pause and think, then resume. Well, the answer is the good old water molecule. It's reacted with the ethene in steam form, and the ethene double bond breaks open, allowing addition of H2O. Phosphoric acid is used as a catalyst. Note that ethanol is the only product in this reaction. The ethene route is faster and more efficient as there is no waste product formed, only the useful one. Fermentation also yields carbon dioxide, which isn't useful to us. But ethene is cracked from crude oil, which is a non-renewable resource, so we won't be able to carry on acquiring it this way forever. Plant sugars, on the other hand, are a renewable resource. The ethene route also requires much more energy. The initial crude oil must be heated to a high temperature, and so too does the water to create steam. The fermentation route requires heating only to 37 degrees.